בנייצ'ר.
אתה יודע למה, קראת אותו. אני גם מתעסקת בנייצ'ר. אני רוצה להגיד לו תודה על המאמר הבאות, אני לא קדימה לזה לכם נייצ'ר, באמת.
אני לא מכיר את הנתונים האלה.
לא, זה תוכנית בקטע של לפתח פידבק, כאילו ולפתח פידבק פנימי, מצד אחד הרבה דאטה שבא ברור שזה מה שכולם
אני בטוח שהיה קצת רעב
כאילו יש אצלנו רוצה להגיש את הפידבקים כאילו כמו
יש שם הרבה פחות סקולה
שזה ממש
תהיה בעיה של רזולוציות גם של הקלסים וגם של כסף והאיסוף של התמונות אבל זה דברים שתרצי אותם אני וג'נליה את יודעת בטח
יש תוצאות
זה בדיוק שהוא, כן זה עושים בסטארט-אפ שתותר מהדגים אני בג'נליה בסדר, יש לזה גם יתרונות
דברים הולכים טוב
היו משפחתי זה שונה כל הפרסס לא עושה שם לבד להיות אחד
כן, זה העקרונות של
אתם עדיין עם העסק
גם מאוד מעניין כזה פעילות עבודה הכי גבוהה מאוד מעניין
זה לא נוצר, יש שם חוויית בידוד בין
לא, זהו, אני לא
קדים,
שלושה ילדים קטנים
זה לא
את לא יכולה לפתח ולהשאיר את זה
לא, אין שום כוונה
אני חושב שלי נראה שיש הזדמנות ודווקא העובדה שאתם משפחתיים
ברור, זה דומה
יתרון
זה דווקא יתרון
זה יתרון מבחינתו
זה יתרון, אנחנו רוצים להיות מעבדה מקצועית
אני אגיד את החברות
הסתכלתי, ראיתי שכן
הוא די טען עליי, זה פעם העבודה של
כן, לכן אני חושבת שבכל מקרה אני אעשה דיסקליימר רציני ששכחתי לו כבר במייל, שכאילו זה הפוזיציה שאני לא מתכוונת לעצמי ללמוד אתה יודע, לספוק את הכפות החשוב במנות
לא נראה שזה משהו צריך להבין בצורה שפשוט
אבל עם great experiment לזה משהו כזה
בדרך כלל העבודות שהציעו להם
כאילו
לא הציעו להם משהו של R&D כאילו אלא אם כן יש מנהלים בסופו של דבר בדרך כלל שיש אחראי עליה אני חושב שזה לא יותר גם מנהל של צוות אומרים טוב, אתה דוקטורט, פרסמת יפה, אתה חכם, אתה מוצלח אין לך ניסיון בתחום, אין לך ניסיון עם אנשים מבינה, זה משהו שהוא כן כאילו זה בעיה נגיד ככה, שזה לא כן, כן אני לא בינתיים אני חושב שכאילו נראה לי המסלול הזה מעניין אותי בכלל אני למדתי שוב נחמד חגום על לוחם
Okay, let's start. So, this is the last time in the foreseeable future that I'm teaching physiology. Next semester, at the end, me to London are going to take over the teaching. Now, in consequence, I'm not going to change the format of the course. It will be like last semester. So, be 3 types of meetings, lectures, regular lectures, mostly by me, also a little bit by others. There will be paper presentation sessions, and there will be advanced lectures. Things that are marked as advanced lectures are not exam material, but you are required to attend. Regular lectures are material that might appear in the exam, but you are not required to attend. And in the paper presentation session, you are also required to attend. Now, please pass a page, mark your names and emails. Okay, I need to know the names of everyone who intends to get credit for the course, because everyone who gets credit will have to present in one of the paper presentation sessions, like last time. Okay, so I distribute you between different sessions, and same rules as last semester apply. In terms of the subject that we'll cover, I'm going to give an introduction to sensory systems in the next few weeks, and then Idan will give a lecture on single neuron computation, models of single neurons, something that should have happened at the beginning of the previous semester, but it didn't. We'll have a very short sequence on somatosensation, because we already talked about it quite a lot last semester, and then auditory and visual. Olfaction will be given by Yoram Ben Shaul. Do you know Yoram? He's at the medical school now. And these two sessions at the end have been on the syllabus for many years, and I never gave them, because there was always reasons to push other lectures next time. In principle, the idea is to, in a way, get back to the lectures I'm giving to you now and see how computational models help in understanding this form of sensory physiology. I don't know if you get the meeting during the ISFN is cancelled, and also the 2 meetings of the week of the ESSEC ICNC meeting, both Tuesday and Thursday. On Thursday, I want all of you to be at the meeting. Okay, that's the day, that's the day where the members of the steering committee are going to give lectures. So there will be leading physiologists who are going to talk. I think it doesn't make sense for you to be here in the class while they are presenting. Okay, so all of you should be at the meeting on Thursday. Also on Tuesday, but that I don't mind that much on Tuesday. On Tuesday, the meeting on Tuesday is going to consist of lectures given by ICNC students who have now faculty positions in Israel and elsewhere in the world. So if you want to see your future, you are invited. But the Thursday session is really going to be very special, something that how to encounter even abroad. So you should be there. Yes. Which? There are the ones that are not for the exam are the ones that marked advanced lectures. They are marked here. Okay. This document will be on the website like last year. I didn't build it yet. Okay, so it will be on the website, and you can. There are 3 advanced lectures. Okay, so these are the ones that are not part of the. Idan will give questions to the exam. Also, one of the comments that I got on the feedback last year was that I'm speaking too softly. So do I? Those who think those who sit at the back. I mean, one student wrote that it was impossible to hear me, that the video did not help because I was speaking too softly anyway.
Yeah, the video doesn't work well without the microphone.
But in class you hear me?
Yes.
Yeah.
If we want to tell you.
But is it important that, do you feel it's important that the videos will work?
Yes.
Yes?
Yes.
Okay, so remind me to put this is the microphone. Remind me to put it on. Okay, I need names only of those who are going to take the course for credit. Okay, so there's required reading. It's always Kandel and Schwartz 4th edition. Okay, there's already a 5th edition, but we didn't get it. I mean, there's a new edition, but that's supposed to be better, but we didn't get it yet. So the required reading are on the syllabus. Okay, so sensory systems. Why do we need sensory systems? To sense the world. Okay, why do we need to sense the world? Yeah, okay, to pass our genes from one generation to the next. Okay, that's okay. So here's a sort of mid-level answer. You know, you obviously know David Marr. Okay, we talked about him when we discussed the cerebellum. Okay, that's so this comes from his book Vision, published 1980, still influential. Okay, so seeing is knowing what is where by looking. Okay, now, does it satisfy you? Is this a good? Why not? Neta. What's the tautology? Okay, so what's the problem? What's the tautology?
Because seeing and looking looks to me the same.
So one thing that I will try to argue here is that they are not. Okay, that in fact this is not explaining what seeing is, but defining what seeing is. Looking in David Marr's in David Marr's view is what happens in the eye, basically. Okay, what happens in the retina. Okay, whereas seeing is the interpretation process. I will try to call the looking part, I will call it sensation, and the seeing part perception. These are bad names, but I need to use different terms for them. Okay, so that's one problem. One issue is this fine distinction between the acquisition of a sensory input and what we do with it. Okay, so that's this is one fine distinction. Why? I mean, other reasons why this is unsatisfactory. What? Okay, so what else you want to?
I don't see understanding what you, I mean, seeing is also understanding what you're looking at.
Okay, so there's more to it than just what is where.
Okay. What is where is also something of perception, not sensation.
No, but that's the definition. Okay, so knowing what is where, that's defining these are the things you need to extract according to David Marr, and this is what it tries to account for. Is start with the image, basically the retinal image, and end with a description of what is where. Okay, that was the task that he set to himself and that he describes in his book. Okay, so
also, so really where we look is also driven by what we know about what we.
Okay, yeah.
Also, you can know by hearing or by somatosensory.
Yeah, we'll get to that in a moment. Okay, so the following set of slides is stolen from Peter Latham from Gatsby, but with his permission, it's okay. So this is basically the problem as posed by Marr. Okay, you have the objects in the world. Okay, they generate a retinal image. The retinal image is not the object in the world; it's a transformation on the object of the world. It's a measurement on the object in the world. And what you want to do is to reconstruct the object in the world. Okay, get back the thing that gave back to gave you the image, and part of the critique that you expressed relative to this definition is resolved to some extent by the way people think about it today. What they talk not about reconstruction of the 3D scene, but recovering of the latent variables. Okay, so what's the meaning of the latent variables? This is the world. Okay, this is the looking. Okay, this is the seeing. Okay, and that's the result. Okay, so these are the latent. Okay, these are the latent variables. So there's more to it than just the house and the tree. There's also evaluation in this case of the quality of drawing of the 4-year-old nephew of Peter, but it doesn't matter. Okay, so this is the problem. The way that people almost invariably try to approach this problem today is by building a so-called graphical model. So x1, x2, and x3 are the latent variables, the causes in the world. Okay, one of them is house, one of them is tree, one of them is bad artist. Okay, now all of them together go through you can write down, you can try to write down, usually it's not very difficult to write down a so-called generative model that will tell you what the retinal image would look like, given that you had a certain amount of activations here. I mean, I'm hand waving. Okay, now this gives you what the image would be if these causes would be, this latent variable would be in the world. Okay, that's usually the easy part, and the difficult part is inference. Given that you have seen an image, what are the causes that that made it? A lot of people are using these type of models. They are all around in various guises. Okay, that's fine. Okay, now we would like to say something about this, how this is implemented in. If this is what the brain does, of course, how this is implemented in neurons, okay, and at least according to Peter Latham's method, okay, we have no clue, okay. Just a moment. So Anna, Orly, Talia, Rotem, Chaim, Jonathan, that's you? Okay. Nice to meet you. Michal, Tamar, Daphne, that's you? Nadav, Neta, Ronen, Itamar, and Ilya. Anyone else? No, just you. Okay, fine. Okay, Okay, so we can take it even further and put, which is very important, and put it in the context of more than just the sensory and perception process. Itamar, this is related to something that you said. So having recovered the latent variables, we'll have a representation of the action that we want to perform. This will get transformed. Okay, that's the story of the motor command that we discussed last year. Okay, so we have a representation of the command, and at the end we have peripheral spikes that activate muscles. Okay, and then we have the motor actions. And of course, the motor actions themselves are going to modify the world. Okay, we kill the bad artist or something like that. Okay, and then this would change the sensory input. What? Okay.
But the motor action might also change the sensory process.
Of course, I mean everything here is also plastic, so the whole thing is very complicated. But the point is, the point of this scheme is to put explicitly the intended action in the same on the same footing as the perceptual processing. We'll see in a moment why this is important. But at the moment, the way that we, when only talking about the sensory system, we separated between sensation, which is getting the information from the environment. Okay, so we have the sensors, eyes, ears, and so on. These are physical measurement devices. Okay, sit usually on the sensory periphery, not always. We'll see it in a moment. Okay, and so in this series, in these first few lessons, we'll discuss mostly basically the interface between the physics and the nervous system. So these measurements are limited, for example, by physical constraints. We'll see a number of these physical constraints. By statistical constraints such as signal and noise. If I have X amount of noise in my measurement devices, this limits the resolution I can achieve. Okay, it doesn't matter why this noise is in. Once I have a certain amount of noise, then this means that there are things I will not be able to measure. By neural constraints, okay, neurons are not just any ideal measurement device. They have their own time constants, space, size, and so on. Okay, so all of these, we'll discuss all of these things. So this, at the end of this process, you have your best representation of the world. Okay, we'll see that it's very partial, it's ambiguous, it's noisy, and we have to take it and get out what happened in the world. So it's good enough for that. Proof is that we survived. But okay, now one thing that we'll see from time to time is that in fact the access that we have to the world through this these measurement devices is extremely filtered and interpreted. Okay, so we can get easily illusions. Illusions is basically when the nervous system tells us things that are different from what happened in the world. Okay, we'll get there. So again, when we go to perception, we are talking about the interpretation, extraction of the latent variables. We talk usually about central processing, so this will go. The sensation will be the peripheral device. Everything that happens while the information go beyond the basic sensors will be already on the way to perception. Okay, and there are two things here, two terms that come up a lot in the recent literature, and that might be important. One, and that's the issue of active perception and perception for action. I mean, they're related to each other. They're not identical, but they're related to each other. Active perception is related to the fact that we rarely perceive the world without doing something to modify our perception. I'm taking this, I'm moving my fingers around. Okay, that's the classical example that there was a psychophysicist called Gibson, who came up with this idea of active sensing. The fact he had a sequence of experiments suggesting that if you put an object on the hand, you can recognize it much less well than if you allow the people to do this. Turned out that his experiments were flawed. Okay, if you give the same amount of information actively and passively, then you get the same level of recognition. Okay, however, so if you do this actively or you move this passively, you will have the same recognition level. Still, we clearly move our sensor around. Okay, so for example, human vision is foveal vision. We really see only on a very narrow few degrees near the center of the visual field, and we all the time move our eyes. So the vibrissa system in rodents, they all the time move their, not all the time, but they move a lot of whiskers back and forth. In olfaction, you have sniffing. Okay. And everyone comes to audition, so what's active in audition? Okay, so people say you can move your ears. There are animals who do move their ears, and cats, for example, move their ears. So blind cats do periodic movement with their ears up and down, which helps them localize sound sources. Most humans don't move their ears. You move your head, which is okay. Nevertheless, there's a subtle. I don't think that audition requires activity in any sense. Okay, I really think that audition is to a large extent passive. I don't know of any convincing examples of a requirement, not the possibility, but the requirement for active exploration of the world by audition. But maybe audition is different in that respect. And perception for action, okay, that's the concept that again, you never perceive things for their own, or you rarely go to perceive things for their own beauty. Okay, I'm looking at my cup of coffee, and I need it. I'm looking at it because I want to pick it up and show you an example of perception for action. Yes. People claim, and again, this is debatable, but it's in the literature, that in a way you have separate systems, separate visual pathways for action and for conscious perception. Okay, so you are more precise, for example, in evaluating the size of objects when you try to reach them than when you just look at them. Okay, so what do we hear for? Ilya, this is, you said we also have other senses. Okay, so this is a different way for defining seeing. This is actually, Reza Shadmer actually said that in a meeting that I attended a few years ago. Okay, so we said, what is seeing? Seeing is, now when you look at this, you see two important features, okay, which are not present in David Mao's description. One of them is the issue of perception for action. Okay, so in order to drink that cup of coffee, okay, and the other one is this issue of predicting. Okay, the fact that seeing, we not, the Reza's point is that we are not constructing the world frame by frame. Okay, we use our previous information about what's out there in order to advance our model of the world a little bit, and the sensory input is used, according to his view, the sensory input is mostly used for verifying that our prediction is correct. Okay, Tali Tishby will tell you that this is the only way by which we can actually perceive the world. Again, this is debatable. I tend to agree with Tali, but it's not everyone does. Okay, so the idea that perception is, in a way, perception is active in a different sense than the Gibsonian sense. Perception is active in the sense that you have the model of the world, you actively move it forward to get how the world will look at the next moment in time, and the sensor input is not used to build it, but to verify that your predictions work. Okay, so let's go to hearing. Okay. What do we hear for? Okay, what's the meaning of this picture? This is Borochov Street in Jerusalem. Okay, that's this house, this building is a school for deaf children. Okay, just near where we live. So every morning there are the minibuses that bring the deaf children from all over Jerusalem to the school. Okay, and on the streets we have this sign. It's not on, by the way, it's not on this. It's not in this place anymore. It's moved. So I took this picture, I think 2005 or something like that, and I'm showing it again and again. And then 2 or 3 years later, Yoav Tal told me, but I went by this place and it doesn't exist anymore. I didn't pay attention to the fact that they moved it by a few meters. Okay, I didn't see the fact that this road sign was moved. Although I'm going past it every day. That's speaking about seeing and perceiving. Okay, anyway, the deaf children need to be, the drivers need to be warned that deaf children are crossing the road. Why? What? It's worse than they can't hear the warning. Okay, it's before even they can't hear the cars. Okay, so it turns out that for deaf children, crossing the road is almost more difficult than communication. They learn, there's a lot of emphasis in this school on communication. So they learn sign language, they learn how to use lip reading and so on and so forth. Okay, but the crossing the road turns out to be dangerous. Okay, they can die. Okay, communication, you can fight with it. Okay, with this, it turns out to be an important part of the reason why deaf people are isolated from society. The fact that they cannot go out to the street. Okay, so here hearing does a job that's very different than the one that we usually think about. Okay, in a way, this goes very well with Reza's definition. Okay, what we want is to build a picture of the world. Okay, in this case, we verify by listening, and in order to cross the street. Why do we need both vision and audition? You almost said that. Yeah, exactly. Audition is omnidirectional. Okay, human vision is very limited. So we have a high resolution sense. Visual resolution at the center of the visual field is about 100 times more, have a resolving power which is almost 100 times better than audition. Okay, we can resolve, in audition, we can resolve about a few degrees, 2 or 3 degrees at the center. In vision, it's about 100 times better, but vision is very limited. Audition is omnidirectional. So course, that's not the only thing that we do with audition, in the same sense that knowing what is where is not the only thing that we do with vision. Okay, so the real interest, I mean, we'll talk a lot about the principles of design of physical measurement systems now, but the real interesting part of the course is actually what happens beyond, for me at least, is what happens beyond sensation, how sensation is transformed into perception. So the classical view, you have the peripheral encoding of physical variables. Okay, that's the emphasis there is usually to demonstrate that it is highly sensitive, that it is limited by physical limitations and not by other limitations, that it has a high fidelity so that you can read out the original values very precisely and so on. And then you have a lot of brain areas that process this information. And the main thing that we do when it processes this information is throw out irrelevant information and get latent variables, so more and more abstract versions of the same thing. Okay, for example, can you see what's wrong here? Who see the two places where it's wrong? Okay, this is an old demonstration that was developed by, I mean, it's repetition blindness, described by, what's her name, the face area woman, Kanwisher, yeah, Nancy Kanwisher, before she started looking for face area. So, in a way, it's obvious what's going on. Okay, the high-level abstraction is the message, okay, and the low-level implementation here is the fact that the sentence itself, and the fact that the is and when repeat themselves is sort of that's irrelevant information with respect to the message. So we are not directly aware for it. The implication, okay, of this type of examples is that we have conscious access to high level of representations, and the information may be abstract. Okay, so you get the things like the Stumpf effect, so you probably know it. Okay, so shout very loud and as fast as you can the color of the letters, not the word. Okay, the color. Okay, so between the red and the green there was a slight delay. It's difficult to demonstrate this because, well, sorry, it should have been in English. Yeah, but also in the red you had to translate. Okay, anyway, for the non-Hebrew readers here, that shouldn't be a problem. Anyway, there's an interference here between the semantics and a low-level feature, which is the color. Okay, you all heard about it. And things like the McGurk effect. You know what the McGurk effect is? Okay, you hear. So look at this is an audio-visual illusion. The sound says something, and you look at the face, which says either that thing or something else, and you hear different things depending on what the face says. Now, there are some good examples of this on YouTube. Look under McGurk. However, you need to be close to the screen in order to see them. When projected on this large screen, then the effect is it doesn't work well. Okay, so yeah, lips and tongue. Okay, so the chapter 21 of Kandel and Schwartz presents the standard model of sensory systems, and it deals mostly with low-level aspects of sensory coding. A lot of it is generally accepted as correct. There are, as usual in textbooks, there's some statements which are wrong. I'll let you filter it. Okay. So we go now to sensation. So from now on, I'm going to talk about, again, design of sensory systems. So how many senses do we have? These are the 5 senses as defined, as usually defined. Okay, so what makes a sense a sense? What makes sense a sense? Okay, physical variable, that's the most obvious thing. So the information you can extract out of it. Okay, keep that in mind. Okay, so first of all, the most obvious distinction between senses here are the physical stimulus that's being sensed. Okay, this gives you a good separation between somatosensation, vision, audition, and smell and taste. Okay, smell and taste, sometimes people call them the chemical senses because in a way they sense the same things. Okay, so what physical variables can be measured? Here's a long list. I'm not going to read all of it, but there's a lot of different physical variables that are that may interest us. Okay, but there are, you said the awareness. I mean, I wouldn't say awareness, but information that can get out of it. For example, in somatosensation, there start to be some problems because the examples here, proprioception and touch, both are mechanic senses. Okay, so both have to do with measurement of mechanic distortions. Touch is on the skin. The proprioception is, we discussed it last year. It's mostly length of muscles, tension in tendons and things like that. That's all mechanical, also mechanosensation. And nevertheless, they are being considered as different things. They are close to each other. Why are they close to each other? Same physical measurements, but in a moment we'll touch on physical measurements that are also mechanical but are not similar. Both go through the same, basically through the same pathways. So both are sensed in the periphery of the body, move on peripheral nerves into the spinal cord, processed in the spinal cord, and so on and so forth. So in that respect, they are processed similarly. But I think that once people become aware of what proprioception is, it's not difficult to argue that it's different than touch. Okay, so people talk about submodalities, different modalities. Okay, so somatosensation in particular has a lot of different modalities. Yes.
Proprioception, as I know it, can be also visual. It's the perception of the movement of your body, so you can.
Yeah, there's.
Visual proprioception.
Okay. Yeah, it's.
Or somatic proprioception.
That's true. That's true. Okay, that's. Classical example is standing on one foot with eyes closed. Okay, so it's easy to stand on one foot with your eyes open. If you close your eyes, then you lose your balance very fast. What? No, but you don't know. Of course, there's also component of balance here, but the thing is that you part of the reason that you fall is because you don't know where you are, so you can't correct yourself.
When the train is
yeah.
moving.
It's true. No, you're correct. I stand corrected. Still, most proprioception is not visual. Okay, you can also proprioceive in the darkness. Okay, so.
You think that you can talk about the higher level abstraction.
Yeah. No. Yeah, you're correct. I'm talking again. I'm emphasizing the periphery. So So what you are saying is that vision is also used for perception in the same sense.
Because you said about abstractions of senses. When you talk about abstraction, then you get...
Yeah, but again, right now I'm talking about the periphery. I'm emphasizing the periphery, okay? We are talking about sensation, not about perception, okay? About the sensors. Okay, so I can wave my hand out of your... yes, fine.
What about the internal sensation?
The?
Internal sensation of,
We'll get there. Okay. On one of the next slides. Okay. So in the five senses, if I look at the what are the physical variables that are being measured, so somatosensation is kind of an omnibus sense. It has many different physical quantities that are being measured. Vision is electromagnetic radiation into limited range of wavelengths. Okay, about 1 octave. Okay, from blue to red, you double the wavelength. Okay, so that's all the range of wavelengths that we are sensitive to. Audition or pressure in the pressure waves in the air, and then you have 2 chemical sensors. Okay, now, time. So even humans have more senses. I mean, again, the most obvious example is the vestibular system, which is sorry about the typo, which measures accelerations. By the way, the vestibular system is the best understood sensory system of all. Okay, what's the computational problem in the vestibular system? What do you measure? We said acceleration. What's the output? Location, position. Okay, how do you go from acceleration to position? What? No, you need initial conditions, but what's the operation you do? 2 integrals. Okay, so we know that the first integral is in the ear itself, is in the peripheral sensor. The output of the vestibular nerve is mostly a velocity signal, not an acceleration signal, and that's because of the physics of the sensor. Okay, it's known how this happens. And the second integrator is somewhere in the brainstem. People know where it is. People don't know exactly how it happens, but they know where it is, and it can be disrupted. And so, in a way, the vestibular system is understood in that sense. Okay, you know what needs to be calculated. You know how, at least where it is calculated. Okay. Then we have all of the submodalities of somatosensation. So even touch is not only touch. We talked about it. We have touch. We have vibration. Okay, we have the proprioception, and then temperature, pain. Not pain. I should have written here nociception. Okay, nociception is the sensory aspect of pain. Okay, and then Chaim, we have a lot of sensory systems that measure things that are that are inside us, not outside us. Okay, so there's this notion of the internal milieu that's part of general physiology, not of neurophysiology. Okay, the thing that the doctors are dealing with. Okay, they want your temperature to be okay, the chemistry of your extracellular fluids to be okay, and so on and so forth. So you have sensors of pH, you have sensors of concentration of all kinds of materials in the blood, sensors for osmolarity. Why is osmolarity important? What? Go to? To the bathroom. That's one direction, yeah. First, yeah. Bathroom, by the way, is mostly mechanical. It's a mechanical sensation from the bladder, but When you're thirsty, yeah, that's to a large extent being thirsty is a measurement of the osmolarity of the blood. Okay, blood pressure. That's very important. So this is mechanoreception, classical mechanoreception. How do you measure blood pressure? Baroreceptors. Okay, baroreceptors. How do baroreceptors work? Okay, it's simply mechanically sensitive nerve endings that are stuck into the wall of the blood vessels. Now, when blood pressure go up, the blood vessel expands, and so these nerve endings are distorted. It's exactly the same as touch. Okay, basically the same sensors. Okay, however, they measure a completely different value. Okay, they measure the blood pressure, and they cause all kinds of changes that we are usually unaware of, but that are, of course, very important for the maintenance of the body. Yes.
Why did you say that these sensors are not? I mean, it's not that necessarily they are not starting this course, but they are part of the central nervous system.
No, of course, but we said I asked how many senses there are. Okay, that's we all the time discussing how many senses. So these are senses that are not part of the five senses. Okay, but they are there. Okay, they are being processed by the central nervous system, usually in a way that we are not aware of. Okay, there are a lot of the loops that are related to them are very much go through the brainstem. Okay, and do things that we are not directly aware of, but they are very important. Okay, let's take a 10-minute break. We'll restart at 10 past. מה? הפסקה עכשיו עד 12, כן. כן. הבעיה היא שחלק A של שנה הבאה יש שוב, זאת אומרת בסמסטר הבא יש שוב פיזיולוגיה סנסורית. אז כי הם מחליפים את הסרט, הם מחליפים את הסדר. זו הפעם האחרונה שאני מלמד. אז אין טעם שתהיה בעיה פורמלית. את תצטרכי לקחת פעמיים פיזיולוגיה ב' אם את רוצה לחסות גם מערכות סנסוריות וגם מערכות מוטוריות. אני לא יודע, שוב, תצטרכי לדבר עם את מוכנה שנדבר בסוף השיעור הבא? אני צריך ללכת שנייה? בסדר? בסוף השעה הבאה, לקראת 1. בסדר? אני פשוט צריך ללכת עכשיו שנייה למקום אחר. הם יושבים שם.
אוקיי.
יש עכשיו... מה, יש שיחת ועידה עדיין בחדר השני?
ב-12 אמורה לסיים.
יש לי עכשיו 10 דקות הפסקה.
אל תגזים, רק 22.
לא, היום זה הוזר 26. וגם האמת היא כשהגענו שם, חשבתי שהתחלנו רחוק. לא, זה חייב להשתנה, זה לא חייב להיות...
Okay, let's continue. Okay, so in addition to all the senses that humans have and that are not part of the five senses, there are also a lot of senses that other animals have and that humans don't in various ways. Okay, so here are examples. Echolocation. Echolocation is not audition. Okay, you shout and you hear the echoes and you compare your shout with the echo and you know things about space. Okay, for example, echolocation measures very well distance. We are very bad. We are reasonably good at knowing direction of sound sources, but we are very bad at judging distance. Okay, echolocation gives information, also very precise information about distance. The vibrisal system, considered as part of the somatosensation, somatosensory systems, but it's very different. Okay, that's we don't have an active hair-dependent sense. Okay, in that sense, the vibrisal system is very different than any sense that we have. What? Yeah, the whisker. Systems that measure air and water motion. For example, the lateral line of the fish. You know what the lateral line of the fish is? Okay, so fishes have a line along their bodies, with a kind of a half-open tunnel that has hair cells in it, like in the auditory system. Okay, and what it measures is the flow of water along the body of the fish. Pheromones. Okay, that's a sub-modality of considered a sub-modality of the olfactory system, but it's different in the sense that its effects on the body, the interpretation of the information is very different than in regular general purpose olfaction. Okay, seeing in the ultraviolet and infrared, electric fields, for example, the sharks that Yossi Arom studies produce weak electric fields and sense their environment by the distortions of these electric fields. Magnetic senses, so this has been just a few months ago there was a big news item about the identification of the magnetic sensor in mammals. Okay, so we don't sense magnetic fields, but there are animals that do, yes.
There are some evidence or claims that say...
The what?
There are some claims that people can sense magnetic fields. For example, certain aboriginals have an amazingly well ability to sense the noise, whatever you do to it. So, is there any evidence for people that lack one basic sense during extremes can really compensate by
magnetic sense? No, I don't know.
Magnetic sense is almost an evidence by Aborigines. For example, whether or not blind people can echolocate better, way better than us.
So humans can learn to echolocate, and there's a lot of this is out there in the literature. Again, search on YouTube. There are these examples of blind people who click with their tongue and use the echoes in order to navigate. Yeah. So people can drive bicycle. Blind people who can drive bicycle while clicking. Yeah, things like that. I wouldn't. there are movies, okay? I don't know how blind is this blind person, and I don't know how much he uses the clicking, but the movie exists, so you can see, you can look at it, you can see. Okay, so humans definitely can echolocate, and I have a colleague who studies echolocation in people, in sighted people. So echolocation is hearing echoes and using echoes to echoes, hedim. So this for sure. Now, sensory compensation and is beyond the scope here, let's say. I don't know about the magnetic sensor. Okay, I never heard about magnetic sense in humans. May or yeah.
Exactly, but nevertheless, you put them in a closed room, you know, circle, without seeing, and then you blindfold them, and then...
I know it works when they're outside seeing the sun.
Okay, let's go beyond this. Orly, fine. Guys, okay, I would like to claim that in fact human audition, cat audition, and rat audition are not the same thing. Okay, that if you are studying a sense deeply enough, you will find that in fact each sense of each animal is different. Okay, some of this is trivial. For example, rodent vision and human vision is not the same. Rodent vision is not foveal, okay, and it is omnidirectional. They see behind their heads. Okay. On the other hand, they have a very limited binocular field. So we use a lot binocular vision a lot in order to judge three-dimensional, at least close to near us, not very far, but within a few meters of us. Okay, the rats cannot do that, but on the other hand, they see behind their head. They see so far behind their head that there's a binocular field behind their head. Okay, the two fields goes like that, so they also overlap on the back. But audition is the same thing. The frequency ranges are different. Rodents don't hear below one, most rodents, I mean the rodents, rats and mice do not hear much below 1 kilohertz, which is, we hear up to about 20 hertz, and these low frequencies are extremely important. You can't understand speech if you cut out these lower 1 kilohertz of the sound. We use interoral time differences to identify location in to do auditory localization. Rats and mice cannot do that because they don't hear at low frequencies. They don't hear at low frequencies, and so on and so forth. So the senses are really species specific. When we say that we study audition and we study audition in rats, okay, we have always to put all the qualifications. We cannot study a certain range of phenomena that are important for human hearing, and on the other hand, rats and mice do things with their hearing that we can't. For example, hearing ultrasound, hearing frequencies that are far above our upper range of hearing. Okay, now, so we discussed what we measure, and I want to go now through some general principles that are common, I don't want to say to all sensory systems, but to almost all sensory systems. This is biology, so there are exceptions. There are always exceptions. So, principle number one, there are receptor cells, and usually the receptor cells, there will be an array of receptor cells, a lot of them, and they will sit on an epithelium. Epithelium is a flat, one cell layer, a layer of whose width is one cell. Okay, so we have the retina, In addition, we have we'll see that the basilar membrane and the organ of Corti where the hair cells sit, the skin, the olfactory epithelium, okay, the lateral line. Okay, so here's, for example, part of the sensory epithelium of the ear. We'll talk about it again. There's a long tunnel which is coiled, forming a snail-like structure. That's the cochlea. And what you see here is a structure that sits along this, this tunnel. So the long axis of the tunnel is this. Okay, this goes for a few kilometers in that direction. Few kilometers. Yeah. Once I magnified the bit of the... It's a few millimeters long. Okay, in humans, I think it's 4 or 5 millimeters long, something like that. Okay, now on top of that, these are the hair of the hair cells. Okay, so you have one hair cell, second hair cell, third hair cell, interval, and another hair cell. Okay, the coiling go in this direction. So these are closer to the inner axis. They are called the inner hair cells. These are the outer hair cells. There's a difference in their function. Okay. All hearing goes through these guys. Okay, the outer hair cells serve for a different function. They determine the sensitivity of hearing. The cells themselves are huge relative to the hair. So here you see the hairs as if they were, I don't know, relatively large, but again, the cells go for about a meter out of the plane of the screen here. Okay, and this goes on. It's a one-dimensional, basically a one-dimensional structure. It goes on in humans with about order of 10 to the 4th hair cells. Okay, retina. So these are photoreceptors. Okay, very densely packed. Note this distance. We are going to talk about that. This is about 2 microns in diameter. So at the fovea, where they are most densely packed, you have 4 or 5 of them in a 10 micrometer distance. Skin, we discussed that already. You have a lot of overlapping, in a way, a lot of overlapping sets of receptors. Okay, now here's the crucial thing, okay? That sensation. Physical variable changes, electrical potential changes. Okay, this is basically it. Okay, now one important thing, it's almost obvious, but nevertheless people make a mistake with that. It's not the physical energy of the stimulus that is transformed into electrical energy. We're not talking about change from one energy kind to another. Okay, so I have a lengthy calculation here that I once made, and I think it's correct, I'm not sure. Trying to compare the energy of a single photon with the energy that's necessary to create the electrical potential that a single photon does. Okay, so this is calculation that you should be able to do based on what you learned in the cellular physiology, the cellular basis of brain function. Okay, so let's do it. Let's go over it. So I went to the literature. One very well-known result that goes back to the 50s is that we can, not one photon, but a few photons can be detected in complete darkness. Okay, so assume that one photon can be detected in complete darkness. The way that people did the experiments is by putting subjects in a room which is really dark, okay, and then using very, very low flashes, and people have to say whether the flash occurred or not, and you can calculate how many photons reach the eye and how many photons. So there's a quantum efficiency, not there's a loss when between the external flow of photons and the ones that are being actually activating photoreceptors. But if you calculate the number of photons that actually got absorbed by a photoreceptor, then you get that 2-3 photons are enough to have a sensation of vision. Okay, so I went to Wikipedia and found that in the range of energies, in the range of wavelengths that that we have, that we see, a photon energy is about 10 to the minus 18 joules. Okay, that's okay. Now we'll see that one photon causes the polarization of a few millivolts. So the question is, okay, the answer is already on the screen, but what is the energy? Okay, where do I waste energy when I want to do a depolarization of a few millivolts? And this is, again, it's basic physiology. I move ions from one side of the membrane to the other, and this motion of the ions costs me energy. Okay, so at the end, I'm going to translate these few millivolts into the number of ATP molecules that I had to hydrolyze in order to move that number of ions from one side of the membrane to the other. So I need to calculate few millivolts of depolarization, how many ions, to how many ions they correspond. Okay? So how do I do that? I need to know if I moved a few if I had a polarization of few millivolts, what's the charge that moved? Okay, so I know how to translate between depolarization and charge. Okay, I need to know the capacitance. Okay, almost everyone with me? How do I calculate the capacitance?
1 microfarad
Per
per
square centimeter. So I need to know what the surface area of the photoreceptor is. Okay, capacitance is basically surface area up to this factor that you mentioned. Okay, so specific capacitance is 1 microfarad per centimeter square. Okay, so for each centimeter square of surface area, I get 1 microfarad. Now, photoreceptors. So again, I went to Wikipedia, and these are not numbers that I know by heart. Okay, I assume diameter of 1 micrometer. It's a bit larger than that. Okay, but length of photoreceptor 30 micrometers. And then I calculated the surface area just of the cylinder without the up and down caps. Okay, so 6 is about 2 pi. Okay, 0.5 is the radius. This is the length, and everything is expressed in centimeters. This is why it's 10 to the minus 4 and not 10 to the minus 6. A micron is 10 to the minus 6 meter. Okay, so I get the surface area, and I know the specific capacitance, and I get that the capacitance of a photoreceptor is 10 to the minus 12 farads. Okay, so now I can calculate 10 to the minus 12 farad times 1 millivolt of depolarization gives me a charge of 10 to the minus 15 coulombs. Yes, Chaim? I'm calculating how much energy I have to spend in order to cause this few millivolt of depolarization. As I said, I need to know how many ions moved from one side to the other, because that's the energy that I pay. Okay, I have then, after these sodium ions moved from one side to the other, okay, I have to pump them back. Okay, and that's where I pay energy. Okay, so I need to know how many ions.
Energy, but you have to pay something for the
No.
chemical.
No, it's not the energy. It's not there's conservation of energy. Okay, let's get to the end of the calculation, okay, and then get back to your question. Okay, now the charge of a single ion is about 10 to the minus 19 coulomb. Again, go to Wikipedia. And I get that I have to move order of 10,000 ions from one side to the other, of the membrane to the other. Okay, so that's the important thing. This is the important result. Now, how much energy do I pay in order to move 10 to the fourth ions, 10,000 ions from one side to the other? Okay, so 1 ATP transfer a few ions, 2-3 ions back and forth. It depends exactly on which ATPase, which pump you are using. So let's say about 1,000 ATPs, few thousand ATP molecules. Okay, now how much energy a hydrolysis reaction of ATP, a single ATP molecule gives me? About 10 to the minus 18 joules, again from Wikipedia. End result, okay, the total energy that I spend to do the few millivolt depolarization is few thousand times. Okay, it's many orders of magnitude larger than the energy of the photon that triggered it. Okay, that's the bottom line.
But if it would be like more than one photon, אם היו הרבה פוטונים שנכנסים בדם מהאוטו דפולריזציה, כאילו אולי זה איזשהו ערך סף של...
Yeah, but I see it. I mean, that's something that happens. Okay, not every day, but because usually you have... but these things happen. So that's... yeah. כן.
אז האמירה היא שלא משתמשים בכלל באנרגיה הזאת, או שמשתמשים במטאבוליזם?
It's not that you use it and need to add... It's like shooting in a gun. Okay, it's not your fingers that supplies the energy to the bullet. Okay, your energy, the energy of your the energy of your finger does is to allow the explosion to occur. Okay, that's basically it. The energy that pushes the bullet is controlled. What you do with your finger, okay, is modify the control signals, okay, and allow this energy to express itself. Or when you drive a car, I mean, you push the gas pedal, but it doesn't. That's not your foot is not the thing that moves the car. Okay, so it's the same thing here. You have another source of energy. It's a metabolic source of energy. Okay, burning the sugars and creating ATP store and so on and so forth. This is where the energy comes from, and that's the energy that will become electrical activity. Okay, you control that energy through the physical stimulus. Okay, so next thing that happens is these changes in the electrical potentials, and they will occur in the sensory neurons, and we call them receptor potentials. Okay, so here is 90% of all psychophysics. You have stimulus intensity, and you have the receptor potentials, and you have a relationship between them, and usually this relationship will be compressive. Okay, the Weber law says that it should go like log, but it doesn't always go like log. Okay, but it is almost always, except for a few important examples, it's almost always compressive. Okay, so if you have at low intensity, small changes in the intensity will give you large changes in receptor potential. At high intensity, small changes in the intensity will give you, the same changes in intensity will give you small changes in receptor potential. This is what it means to be compressive, that it is compressive. Okay, the log law is approximate מכווץ, טרנספורמציה מכווצת. This. Okay, so the same change here gives you larger changes here than the same change here. This is, okay, compression. Yeah. Now, a lot of sensory physiology is about how these changes occur. Okay, so there's a lot of different mechanisms, but not an infinite number, that couple between the physical stimulus and the electrical change. In some cases, you have a direct mechanical coupling between the physical change and the channel. In hearing, I'll show you an example. You might have a ligand-gated channel, so this is good for the chemical sense. If what you sense are chemical compounds, you can couple the electrical changes with the chemical compound simply by having a channel that's sensitive to it. So, for example, one of the best-known examples of these ligand-gated channels is the one that has to do with harif, with hot taste. Yeah, these TRP channels. Did you hear Alex, what's his name? Winstock? Talk about his... Yeah, exactly. Yeah, that's the channel, okay? It's a ligand-gated channel that is activated among other things by the material that gives us the sense of harif. Or second messenger cascades, the most famous of these is vision. Okay, let's look at here is audition. So these are the hairs. We saw the hairs. Okay, the hairs comes in multiple rows, and you have small hairs and larger hairs and taller hairs and taller hairs. The channel sits on top of the hair, and it's connected with a protein wire to the next hair in the next to it. So when you push the hairs in one direction, this wire gets tense, and it causes a change in the structure of the channel. It opens, and ions can flow in. Okay, this is well known. I mean, this is. I mean, it is a drawing, but the evidence for this is overwhelming. People saw these things. They can disrupt them. This causes a disruption of the transduction. Okay. The one thing that people don't know is whether the channel is on that side or on that side. Okay. The channel could be as well here, and the wire going in that direction. So these people don't know. Okay. This is based on one model, but other people believe that it's on the other side. What? The wire is made of? It's protein. It's like a very thin hair attached to the thicker hair of the hair cell.
What is the size of
So the size of what? The whole thing here is less than an order of micron. Okay, but you have a few hundred hairs on each hair cell. Okay, these are tiny structures. As I told you before, when we saw the hairs themselves, here, when we saw the hairs, okay, the hairs are small relative to the cell. The cell itself would be order of a few microns, let's say 10 microns, and this is a small fraction of its size. Okay, so understanding the hair cells, the hair of the hair cells, the behavior of the hair cells is already you are working with structures that are composed of few hundred proteins. Okay, they are very small. On a single hair cell, there are probably 100 transduction channels. That's part of the reason that the transduction channel in audition is not yet being identified. There's so little of it, so little material to work with. Okay, so this is now vision. So it's a second messenger cascade. That's very complex. Okay, I'll try to take you through it. So the crucial thing, the crucial material that open and closes channels in photoreceptors, in mammalian photoreceptors, is something called cyclic GMP. Okay, cyclic GMP is the ligand that causes channels, internal ligand, inside the neuron. It opens and closes the transduction channels. Now, so whether the channels open or close depend on how much cyclic GMP there is. Now, cyclic GMP is produced by an enzyme called guanylate cyclase, okay, which is not under light control or not directly under light control, and it is broken by another enzyme which is called phosphodiesterase, and the phosphodiesterase is under light control. So you produce cyclic GMP at a fixed rate, basically, but you break it at a rate that depends on the amount of light. Okay, and that's the way that you control the amount of cyclic GMP in the neuron. Okay, now, the phosphodiesterase is usually is inactive. In darkness, it is inactive, and in order to activate it, it needs to be associated with another protein, which is called the G protein. Okay, now the G protein, in order to be available for activation, has to be associated with a GTP. Okay, to get a G protein with a GTP, you need to have a rhodopsin molecule that got a light. Okay, the rhodopsin molecule, the activated rhodopsin molecule, connects to an inactive G protein. It's a G protein with some other subunits that make it inactive, connected to a GDP. Okay, the interaction between the rhodopsin and this inactive form of the G protein causes the release of the active form and the exchange of a GDP by a GTP, so you get now the active form. Okay, now each rhodopsin, once it's activated, can do this for a number of can activate a number of G proteins. Okay, and they activate the phosphodiesterase. Eventually, the G protein hydrolyzes the GTP. Okay, so it goes back into the inactive form. That's part of what stops the light. That's not the only thing that stops the light reaction, but that's part of it. Okay, so that's now note that when light comes on, I have a reduction in the level of cyclic GMP. Photoreceptors are depolarized in darkness, and they stop being depolarizing when the light comes on. I mean, vision is upside down in many respects, but that's one of it. One of the ways in which vision is upside down.
Why is it? And how is the depolarization?
What?
כאילו, איך כל הריאקציה לכם מתקשרת לדפולריזציה?
So if you have light, you activate rhodopsin, you reduce the amount of cyclic GMP, the cell body goes through actually not depolarization, hyperpolarization. It goes down. In mammalian photoreceptors, light causes a hyperpolarization of the photoreceptor. When the light goes off, when the light reaction stops, it goes up to higher potentials. So you have the receptor potentials, and then the receptor potentials causes spikes. Spikes can occur in the same neuron or in other